Vice Principal of Science and Engineering, and I get the opportunity to wear the principal's gown. It's a great pleasure to introduce John. Normally when you're asked to introduce an inaugural lecture, you can sometimes read. In John's case, I think it's CV, right? I'm not going to attempt to read out the titles of his papers, but this did tell me, looking at the papers and looking at John's career, showed the breadth of his interests and the enormous respect in which he's held by his colleagues. John grew up in Monmouthshire. He studied botany at King's College London and gained a PhD there in 1975. He then went to study at Oxford, picked up a PGCE along the way, worked in the University of Illinois in Urbana-Champaign for a while, then went to Warwick where he worked on photosynthesis. The work he did there, I'm told, is now textbook material on photosynthesis regulation. I'm delighted to see somebody connection from those days. Tells you a little bit about the breadth of his work, the intellectual interests and companions have picked up along the way. After Warwick, John had, by then recognised as a talented researcher, had several choices of career move. He could have stayed there on an SRC 5-year research fellowship, but he instead chose to move to Leeds. From Leeds, he moved on to Oslo and then Sweden as Professor of Plant Cell Biology. He worked in Sweden for 12 years before coming to Queen Mary in 2005 as Professor of Biochemistry. At Queen Mary, he's worked with a very strong group we have in this field and set up his own group. A number of his research students and research workers are here today. And moving into the area that's becoming one of the main focuses of our study, biological and chemical sciences, photosynthesis and the impact of photosynthesis. We'll get some demonstrations today. The impact of photosynthesis on our work and energy. People have told me that John is a serious person, a serious scientist, known for his philosophical interests as well as his direct scientific research. Somebody said to me, well, he sounds like a conceptual scientist. He doesn't see the science as a box of ideas or a box of facts or a box of techniques. He thinks of science as a set of ideas. People also tell me that he knows how to relax, apparently by listening to music late at night, listening to loud music very late at night. He told me that. My spies get everywhere. But it does give me great pleasure to introduce John's inaugural lecture, Thinking Outside the Green Box, and to congratulate John on being here today to present his lecture as Professor of Biochemistry at Queen Mary.
Well, either it works or it doesn't. Thank you so much, Professor Martin, for that introduction, setting up many preconceptions that I shall cheerfully dispel in the course of the lecture. I'm grateful to stepping in for this, and I'm grateful to your attending. Professor Martin, Professor Heathcote, and the members of the faculty, ladies and gentlemen, the first thing is a slight administrative thing. I don't know whether you've checked the location of the nearest fire exit. We have several demonstrations here. As on entering an aeroplane, I advise you to go two behind. We don't need the slides because you'll just step down into the courtyard. Fire extinguishers, I always look for fire extinguishers. you never can be too sure, because the worst could happen at any time. The kitchen metaphor will return in this lecture. If the worst should happen, and I'm not saying it will, don't give nervous disposition, this is getting serious. What do? Do you email the central administration? There might be reasons for doing that. There might be reasons for not doing that. But hold that question, if you will, because I think the answer tells us something about cell evolution and photosynthesis and many other things as well. I really have to start with a primer. I have in this lecture 3 primers, which most people here will find trivially obvious and boring, and some people won't, and if you don't, you have to know this. Kitchen chemistry, like where the pan catches on fire. Reduction, in a chemical sense, means gaining electrons. Oxidation means losing electrons. It's Oliver Morton from Nature. Welcome, Oliver. Author of a wonderful book called Eating the Sun. Oxidation, losing electrons, and reduction and oxidation together. If you gain an electron, you got it from somewhere, and you got it from something that's become oxidized because it's lost it. It's that simple. Electrons are truly elementary particles, I am told, that is to say they are not made of anything else, unlike protons, neutrons, and everything. And moving electrons means a reduction and an oxidation coupled together, which is shortened to redox, which is not in fact a brand of bath salts. It is simply short for reduction and oxidation. There is this molecule called ATP. There is this thing that moves all around us that we use and take for granted and incorporate into our language and other people called light. What is light? Light is radiant energy, electromagnetic radiation. Light is physics and chemistry and biology. I have a small demonstration for which, first of all, let me just remind myself of the next slide. Yes, the next slide is deliberately nothing. Light has some surprising properties. What colour is this? Anybody? Green. If I switch on this light and we observe it from the side, what colour is it? It's strawberry. This is a chemical compound called fluorescein, which absorbs blue light, and then it either, if you look at transmitted light or reflected light, you see it as green because you see the yellow and red, but you also see a touch of yellow around the edges because it absorbs blue light and emits it again as fluorescence. So light can be interact with matter, it can be absorbed and emitted again straight away. If we take chloroplasts isolated by Sujit Kukuviso from a pea plant, looks like this, earlier today, isolated, this is the green box, incidentally, the first green box is the chloroplast. Looks nice and green, you can study its biochemistry. There's nothing very surprising about that, it's sort of turbid and green. But if you take the same quantity of chloroplasts isolated from the same quantity of peas, and you put it in acetone, and perhaps the background is not brilliant. Can you see a sort of reddish hue to this? The red is light which is being absorbed by the chlorophyll and then emitted again as fluorescence. Red fluorescence. It's not quite as dramatic as that. Let's set it up. So those are unusual properties. Light is that's physics. The chemical reactions, as I mentioned, include oxidation and reduction. This is well known to school children everywhere for measuring the vitamin C content of, for example, cabbage. And it's easy to do spectrophotometry for this because this is a pleasing blue colour in its oxidized form. But it quite readily takes up electrons, becomes reduced. That's many face lights on the to get the effect of this one at least. And we simply tip it into a beaker of some ascorbic acid, so this is magic trick number 1. Again, it's quite good for doing magic tricks, isn't it? I'm used to this idea. No, it's blue. No, it isn't, because it's become chemically reduced by the ascorbic acid in the vessel. So electrons went, a lot of electrons went very quickly from the vitamin C in the flask to reduce the blue dye in, as I call it, in. So that's an oxidation reduction reaction. You might need a little more convincing that light is involved in chemical reactions. And but it's true. I have here a beaker which is solid ATP, conventional ATP. And I chip into it, somewhere in the green, what is the magic spell? What is this? Some luciferin and luciferase. And we swirl it around, and the energy in the ATP is used by this enzyme from extracted from fireflies to make light. Can you see that? The firefly doesn't know it's doing photochemistry, but it is doing photochemistry. And the reason it's doing it is to attract a nice, attractive female firefly. It's a signal, it's advertising. There it is, that's what we're going for the rest of the lecture. We can put that later. So, we can regard light as a chemical. Einstein knew that in 1905, although you don't ever molecules or atoms that so that's primer number 1. I hope you didn't mind that. Primer number 2, that was kitchen chemistry, this is kitchen physics. Energy has many meanings in press and on television. But really, energy is the capacity to do work. Let's take work as a given for a moment. And you measure energy as the capacity to do work, therefore work and energy are the same thing. They have the same units, they have the same dimensions, and you can measure them in joules or calories. I sort of pick calories actually, but the conversion factor is very simple, as my physics teacher got me to explain to the whole class by heating up some red shots by moving it around for half an hour, because I talked by the way. It's 4.2. Ladies and gentlemen, Dr. Fanny's Miss Ellis. Well-known neurobiologist.
Some must think you're talking of 7.
What is power? The principle of Dr. Well, I'll tell you what power is. Power is simply the rate of doing work. That is the rate of using energy. And power is measured in watts, about which we shall be doing much more soon. Watt is a watt. A watt is 1 joule per second. So if you use up energy at 1 joule per second, that's a watt. If you do work at the rate of 1 joule of energy used per second, that's a watt. Light is radiant energy. Light is physics and chemistry and biology. And at this point, I want to demonstrate that not only can you get light from a chemical reaction, you can also use light directly to generate electricity. And that's a matter of demonstration, isn't it? What we have here are 8 100 millivolts, no, 200 millivolts, 100 milliamp photovoltaic cells connected half in series, half in parallel. And you simply shine a light on that, and it drives enough electricity to run a little electric motor, which gives us a very nice reverse wind turbine. Instead of using this wind turbine to generate electricity, we can do it actually. You can blow on it and connect your overmeter and generate a specified amount. Can you see that? And it's not, there's no magic of battery or anything. If I turn the light off, it stops. If I turn the light on, it goes. If I put my hand in the way, it stops. I take my hand away, it goes. Okay, so light is directly making electricity. What's this got to do with biochemistry? Well, actually everything. An awful lot. Huge amount. Let's move on. This is a cyanobacterium. They used to be called blue-green algae. They are the most wonderful organisms you could possibly think of. Anya Nenninger from Conrad Murray's lab gave me this. It's not amazingly dramatic on its own. You can come and look at it later. It's genuinely blue-green. And it happens that G.E. Fogg, professor of botany at Westwood College, it's Ralph Stanevsky, ladies and gentlemen. Welcome, Ralph. I never thought I'd have to tell you off. I'm very disappointed in you. This is a cyanobacterium called Anabina cylindrica. It's made of filaments of single cells which contain chlorophyll, which absorb light, which fluoresce. You can measure that. You can tell a lot from looking at that. And it also has heterocysts, which are specialized cells which use the energy from sunlight fixed in the other cells to fix nitrogen out of the air, which is a very special and dangerous and complicated chemical trick which only bacteria, because they're plant, can do. We can't do it, plants can't do it. They only do it if they get bacteria working for them to do it. And this was a watercolour painted by G.E. Fogg, done by G.E. Fogg in 1946, and I used it for the flyer, courtesy of the events office. And the original was owned by Mrs. Brenda Tate in our department, and I thank her for that. And also Ray Conrad, who opened the scan for a number of the other technical devices you will see today. These are chloroplasts, I told you already. This is the green box. And what I want, each one of these is a green box. Chloroplast is simply Greek for green box, if you haven't got it yet. We'll be talking about other green boxes later. There's one here, for example. Unless it's a blue green box, I'm delighted to say that. Even better, a cyano box. And this is a scanning electron micrograph given to me by John Neal. He got it from someone else. It's just like electron transfer, very graphic. He apologised for this being a false colour SEM, and I thought about this a lot. In other words, the scanning electron micrograph doesn't give you colour; it gives you grayscales, intensity, not quality of light. And what you see here, it's irritating they didn't put a scale bar. And I invite you to consider that each one of these green things is about 10 micrometers, that is 1/100th of a millimeter in diameter, from which I calculate, since it's maybe 100 of an inch length, which is about 1 millimeter of the surface of a leaf, and they clip off the bottom, which is part of the plumbing system that keeps these cyanobacteria supplied with water. And what I want to put to you is chloroplasts, which make all our food and produce all our oxygen from light energy, are descendants of cyanobacteria. And in fact, in many ways, you can think of them still as colonies of cyanobacteria that have occupied a large superstructure that keeps them supplied with water. Here they are. From the science photo library. So this is the green box, this is the chloroplast. It's rather small, 100 millimeter approximately. We'll go on to some rather smaller things. Between that level of size and this level of size, there are lots and lots and lots of interesting things that Conrad and Professor Epstein and other people are interested in modeling, because membranes in which these things contain the fluid, and the fluidity allows mobility. But this is a very nice picture from a book by David Goodsall, published in 1993, before anybody really had a clear structure of what deep down inside the chloroplast's energy converting membrane looks like. This is a membrane. What it has, and the scale here is it's not a microscope picture, it's not an ambient picture, it's imagination plus science, and an artist to make a cartoon. The length of one side here is 10 nanometers, 100 angstrom units. That's 1/1000th of the diameter of the chloroplast in the previous slide. And what we see, first of all, is that this molecule ACP, which we know contains energy because we use it like fireflies to produce light, is being made from a molecule of ADP, a diphosphate, plus a phosphate group, and this ATP, which contains all that energy, is being made as a result of the discharge of a gradient of hydrogen ion concentration. So this artist, David Ursula, has such a fertile imagination that he can cheerfully draw us one hydrogen atom. That is 1 angstrom in diameter, ladies and gentlemen. What we see here is that there is an electrical circuit inside this membrane. There is oxidation of water, removing electrons from water. Remember I said oxidation is removing electrons. That gives oxygen. That's where all the oxygen in the atmosphere comes from. This is an essential reaction. We could not live without it, you know this. Light is then drives in a little photovoltaic cell called a photosystem, a flow of electrons. There's a connecting thing which does a lot of conserving the energy to protons to make ATP. And then there's another photovoltaic cell which uses light energy to transfer electrons which would not otherwise move and reduce a final electron attack called ATP to make ADPH. All a bit technical, but this I think is a very good accurate cartoon description of what we know. And I'm not going to talk in any higher resolution about molecular structures. The electron microscopists worry and agonize about the false color in scanning electron micrographs. Nobody worries about false molecular graphics. Nobody really believes an oxygen atom is red or a nitrogen atom is blue. So good sort of stuck to black and white at this scale. In what way are these photovoltaic cells? It's very simple. They have positive terminal and a negative terminal, and when you shine the light on, you create a potential difference between one side and the other, and that potential difference moves electrons through the circuit from this point where electrons start to this point where the electrons end up. The consequence of that is plants can fix carbon dioxide to build up their own matter, and the consequence of that is they release oxygen as a waste product, a toxic waste product, as the oxygen that the animals breathe and rely on. And notice that these two photovoltaic cells are connected electrically in series. This is important, connected as well. And this was realized before when we put it in the context of the membrane by Hill and Bengal in 1960. Great pleasure to see Derek Bengal here. Such a pleasure to have him here. One always has to sort of go back and dig in and find what a sort of brilliant insight this was, to realize that if you plot the electron transfer sequence of photosynthetic electron transfer on a scale of potential measured in volts, you arrive at the conclusion that light is used in two different places. One would be called pigment system and photosystem 1, the other photosystem 2. And the series connection proceeds like this, and in some way that series connection couples electron transfer to ATP synthesis, and I'll tell you a little bit about that mechanism. All this is wonderful, and every slide here has a whole lecture behind it, and I have to take control and not get digressed. So plants do that. They do that because they have captured cyanobacteria, which they've kept within their cells for about 2,000 million years or so. The cyanobacteria, or blue-green algae, still do that as well. They contribute to the oxygen in the atmosphere. And the oxygen is taken up in little membrane-bound organelles, which also were once bacteria, which is very clear, called mitochondria. And this is a cover from Nick Lane's book, Power, Sex, Suicide, Mitochondria and the Meaning of Life. He said he couldn't get a babysitter tonight, so he sent something like this in Congress. It's a wonderful book. And that's where the oxygen is used to make the ATP that the firefly uses to attract a mate, that I use for standing here giving a lecture, that you use at the rate even of 100 watts, simply by sitting there. Even if you've fallen asleep, you're running at 100 watts. Peter Rich calculated this, not the falling asleep part. Nature. Peter is here. I've gone through that preparation right, and I've tried to do the same for photosynthetic systems. Let's see where we get to. Now the other clue to chloroplasts and mitochondria which make that ATP in animal cells, as well as in plant cells, a clue to their ancient evolutionary ancestry of bacteria is that they have their own genes. They have their own chromosomes. This is amazing. People didn't like this. They wanted to argue it away and say, no, it's bacterial contamination. There are genes there, but it's an artifact. But no, it's perfectly clear that both mitochondria and chloroplasts indeed contain genetic information independently of that of the rest of the cell. And the inference is very clear. They got that from the bacteria that they once were, free living bacteria. Given the fact that they were once bacteria, and 99.9% of those bacterial genes are either being lost or aggressively acquired, what's a hostile takeover by the cell that now contains them, why are there any genes at all left in mitochondria and chloroplasts? Well, the wonderful textbook, The Molecular Biology of the Cell, still in the latest edition, is honest enough to say we do not know. Many other textbooks waffle endlessly for pages about hydrophobicity and strange forces darkly at work. But the molecular biology of the cell says, why do mitochondria and chloroplasts have their own genetic systems? Text, why do mitochondria and chloroplasts require their own separate genetic systems when other organelles which share the same cytoplasm, the same cell structure, do not? The reason for such a costly arrangement is not clear. We cannot think of compelling reasons why the proteins made in mitochondria and chloroplasts should be made there rather than in the cytosol. Now, I have an answer to this question, and I'm going to tell you what it is. That's quoted from our textbook. My proposal, I gave in 2003, this little acronym CORE. I thought it was there's a classically derived things like the isotopic hypothesis, in other words, they should be in the same location of the cell. People pointed out to me that you get the anti-nuclear campaigns in your laboratory at the moment before that. So I thought core would be quite good. It's supposed to indicate an expression of appreciation and core. And it says this, it really stands for co-location for redox regulation, electron transfer, oxidation reaction, redox chemistry, controls expression of genes for proteins directly involved in or affecting a redox reaction. So all those little things plugged in the membrane, cheerfully accepting an electron and passing it on and storing the energy in some way, these are proteins. Proteins are made by genes as a result of the expression of information in genes. And my argument is there's something so special about this redox chemistry that it is vital, and it always has been vital, that information about the electron transfer reactions is communicated directly and unconditionally, without asking permission from the nucleus, to a special subset of genes. The control requires co-location of these particular genes with their gene product. It is indispensable. It's required for life for these sorts of cells that we are made in plants and others. And this control operated continuously throughout the transition from being a prokaryote, that is a bacterium, to being a eukaryotic organelle, that is to say a mitochondrial chloroplast. In other words, this thing works. It was always necessary for life. It was simple, it was effective, it was robust, and as the saying goes, if it works, don't fix it. And that's why chloroplasts and mitochondria to this day still make their own decisions on the basis of information that only they have, environmental changes affecting the rate of the redox reactions that they use to harness and convert energy. So I first logged that out in a sort of detailed way in the Journal of Theoretical Biology, which is all this stuff about one of the serious and philosophical comes from, I suspect. Certain people in the Journal of Theoretical Biology will actually run. Well, not of sound mind, but you knew that anyway. And then 10 years later, there's so much happened in the following 10 years, it really began to look like there was something serious here. And we have philosophical transactions of the Royal Society from which some of the slides come. This is from the 1993. This is our bacterium. So cast your mind back 2,000 million years. It can be a cyanobacterium, one of these nice blue-green things. It could be the bacterium that gave rise to our mitochondria. It's happy. It's got energy transduction system, of course, to make ATP. by moving protons back to a membrane after they've been pumped across the membrane by electron transfer, by redox reactions. And just to stand for different classes of proteins, we have proteins A, which are part of this energy transmission machinery. Proteins B, which are pretty rare if you like, name your favourite protein. Glycerol 3-phosphate dehydrogenase is pretty much protein. Greatly underestimated protein. Protein C might be any membrane protein that wasn't doing this critical energy transduction process, chlorine, some drug receptor, or various different evolutions of effective drug receptors. And suddenly, oh yes, and so all these proteins come from all these genes. It's one gene, one protein. Gene A makes protein A, gene B makes protein B, gene C makes protein C. Now what I've been saying is that the information about what protein A is doing is so important to that bacterium that it has some kind of sensor to send information back to the DNA to switch genes on or switch genes off for that particular protein. So gene A is very tightly controlled by the function of its gene product, because the function of its gene product is A, essential for efficiency in energy conversion, and B, absolutely catastrophically a bad thing if it gets it wrong. So protein A has to be present in the right place, in the right quantity, at the right time. And the way to do that is for it to send information about its function back to its gene, to switch its gene on and off as required. The rest, yep, they receive orders, they get switched to, but it's not quite that important. So, what happens? This bacterium doesn't know it yet, but it's just been eaten. Eaten is slightly the wrong word, because in some way this bigger host cell decided not to digest its symbiote, but leave it there for a while and see what happened. And what happened over enough time is that copies, like copies of files from computer disks, the copying process occurred at high frequency. People didn't like the idea of the high frequency, but they do it, but it's demonstrated experimentally by Jeremy Timmis and others in Australia. And regeneration, a field of wheat, every individual has got a couple of genes that arrived since the last generation in the nucleus from the compound. And the copying gene A, you can make a precursor in the cytosol, the main part of the cell. That precursor can go into the chloroplast, or at the endosymbiote, you might still talk it. You throw away the garbage, and you make a protein A that's indistinguishable from the protein A you made from the original gene. I won't labour this point, I think you can see where we're going. Sorry about the bitmap graphics, I drew it in Canvas years ago when I really enjoyed computers, but it serves the point. Gene B makes precursor protein B, is inserted into this compartment, makes protein B just as good as if it came from gene B in the bacterial endosymbiote, and so on. Now, we have a mess. We have redundancy. There are too many people hired to do a small number of jobs here. I think Dick Cavett said to me once, it seems like a rationalisation of overstaffing. I think that's right. Yeah, I think that's entirely what it is. Except there is now a division of labour. You don't need all these two different copies. So they would be making proteins, there will be a sort of coherent way of regulating things. So what happens is there are good reasons for getting rid of the original bacterial copy of protein of gene B and gene C, making protein B and protein C. Why? Because the nucleus is a nice, tidy, sleepy, dusty, stable place to keep information. It doesn't do anything. It's an archive. It's a hard disk. It just sits there, and the nucleus occasionally wakes up and gives you a bit of information. But it doesn't do anything interesting or important like converting energy. It's sort of a repository of information that might come in useful one day. So that's a good place to keep information. This is a bad place to keep information, as we shall see, because this chemistry degrades information very rapidly. It's a crazy place to keep genes. Now what if we do the same for gene A? No problem. Protein A is just as good, like all the other proteins, if it's made this way. But what have we lost? We've lost that feedback. That all-important key to being efficient and to being safe in the way that you convert light energy. So you don't lose gene A in that compartment. you lose GA from the nucleus because you don't want that redundancy. And so for the redox-regulated genes that encode the key proteins, you keep the DNA where it wants. And this is the bioenergetic organelle, and this is what I would argue we see today. For example, when we look at this chloroplast chromophore membrane, this is false colour again, and ask, well, where is this particular protein made? Green from the chloroplast gene inside the cyanobacterium that was, or brown for a gene making a protein that's important for the nucleus. So this is what you find. Broadly, the same pattern, the same proteins. If you have these photovoltaic cells or photosystems have at their core a photochemical reaction set to make proteins that are always encoded in the chloroplast DNA if they exist at all, without exception. Anybody knows an exception to that, please tell me. And the reason is, I would argue, that these reaction set proteins have to be regulated unconditionally, rapidly, for efficiency, and because they're catastrophically dangerous if they're present in the wrong quantities and at the wrong place at the wrong time. And so, again, the explanation, I would argue, is that here's the bacterium. We call this a cyanobacterium. It could be Professor Fogg's Anabina cylindrica. In fact, there's evidence that filamentous nitrogen fixtures closely related to chloroplasts. If you take it out in the molecular biology evolution, this is going falling off. Let's look at this way. It's very embarrassing. Something here drapes to the shoulder. And here is the redox regulation, which you still find in the bacteria. And in the course of evolution, most of the genes ended up here to make the brain proteins that were important as precursors, but not all of them. Some had to be retained. And this is why I think we have genes in chloroplasts and you see in the mitochondria. So this is all very well. The phenomenon is there. We've demonstrated the phenomenon. Nature paper in 1999 by Tom Fanshley in London in my laboratory. How does it work? Well, quite recently, here in Queen Mary, Sujit Pudivito has inspected closely the complete genome of a small and unprepossessing little weed called Arabidopsis theriana. Shall I get some lights? Can you tell me? I can't see the lights very well myself. This was the first plant, and it was chosen deliberately as a model to have its entire genome sequenced. In principle, one could write a chemical structure of all of its DNA. One could, in principle, you need quite a lot of paper and a ferocious memory, but of course it's all on computer files these days. Sujit had a very, very, very good look with this evolutionary hypothesis in mind at the genome to see if we could find a bacterial sensor which might be functioning to be imported into the chloroplast to convey that information. He found it. And it's chloroplast sensor kinase. Do come in, please come in, plenty of seats. Shall I, it's Angelica Stoliver, who will first of all take off her backpack. And it allows me a sort of breathing space. And I'm sorry, I can see you. Very welcome. It couldn't have been 10 past 7. Histidine sensor kinase, and it's homologous, that is to say, has a common origin with a histidine kinase called H2 of cyanobacteria. It is a redox sensor. It's a finger you've got dipped in your electron transport chain to feel the temperature, if you like. No question. And that's Sujit, and we found it, and there it is. And this is an experiment done earlier this year by Tony Kavanagh in John Gray's laboratory in Cambridge University, in which he took Sujit's gene, he cloned it, he made a whole lot of it, and he linked it up with a fluorescent protein, looking actually quite a lot like this stuff, except it's a protein, and this isn't. And he looked down a microscope, and he saw the red fluorescence. Remember the red fluorescence? There's a little bit of red in there. This is the red fluorescence of the chlorophyll pigment in chloroplasts in tobacco leaves. And then after he transformed the cells, here is one cell, here is another one, forming the guard cells of the stomata, the modes through which leaves breathe, leaded carbon dioxide. He found that the tungsten particles containing the DNA with Sujit's protein kinase plus a fluorescent protein tagged on to let them know where it had gone, transformed the chloroplast. The DNA went into the cell, it made the protein went into the chloroplast, and you can see the chloroplast lit up green in this cell, which had been transformed, and not in this cell which hadn't. By chance, one tungsten particle had hit this cell and not that one. So all chloroplasts are there, you can see their red chlorophyll fluorescence. All the chloroplasts are here, and in this cell, the protein kinase has been taken up, and you can see the chloroplasts. And if you overlay the red and the green, you get orange, which is a very clear indication that this protein has been taken out. And this is an acknowledgement, since this is not yet published, despite 6 months of ferocious argument with dense editors of important scientific journals. The redox sense of kind of village, it will be soon. It couples photosynthesis to gene expression of chloroplasts. And here are the names, and we have a number of other names associated with this, with some help from Matt Rogers, who was a Queen Mary with the exeter with Mark Van de Giesen. We find that this gene, for this all-important protein, is everywhere in green plants. Green plants, bryophytes, mosses, liverworts, vascular plants. Actually, that means green algae. I think that chlorophytes is all of this. It has to include all this. This is the smallest known eukaryote, Ostracopus, I believe, has one histidine kinase. This is it. Diatoms, red algae, you find on the seashore, and of course the cyanobacteria and chloroplast came from, all have this protein. It's indispensable. I call this protein in conversation. It is, for my theory of molecular cell biology, the Higgs boson. I've been saying that protein is there since 1993, and people have been saying, John, you're a load of hot air, relax, come out here and talk about something else, or something like that. But it bloody well is there. It has exactly the properties that you would predict. Even from that 1993 slide, it's possible to say this must be here. And this gene, of course, is in the majority category of genes that could perfectly well afford to move to the nucleus of the cell in evolution. So you know the Higgs boson, they're building this thing in Geneva called the Large Hadron Collider, from which I learned from Christopher Llewellyn Smith, that if we understand matter, there must be a Higgs boson. So we better have a good look at it. Don't we think so? I think so. If we understand eukaryotic cell evolution, there's got to be a chloroplast sense of kinase, and there is. And I think that's the need. So here's Sujic took that figure and we drew it. Here is where the information comes from. Chloroplast sense of kinase is modified when the information comes through the electron transport chain. It sends signals back to the chloroplast DNA to say switch this gene on or switch this gene off. Same is true for mitochondria. I have another lecture on mitochondria. You won't get it, there isn't time. But there's the same hybrid arrangement of proteins that are made from mitochondrial genes and proteins that are made from genes in the rest of the nucleus and then imported. And I think the same explanation applies to mitochondria, and then we have to go completely happy. Hooray. And we're similar to evolution, so that's wonderful. In contrast, signal transduction people, I have any people can I offend in one lecture? They send you emails wanting to buy for thousands of dollars antibodies to the latest set of obscurely named signal transduction components. This is probably, I can't even read it, you can read it over like that, it's back there I'm sure. But it goes on and on and on. I mean, it looks like chemistry in the sense that there are arrows between things. But it's a poor life for a decent protein. There's nothing wrong with proteins. They're good, honest proteins, and they're the products of good, honest genes. It's the management structure that's totally wrong. This poor protein is required to send a signal to the nucleus saying, I've just got a signal from somewhere else. That's all it does. That's not a very inspiring job description. And then the receptor says, well, I've just got a signal from you, but how do I know you got it from him? I better have another protein to check where you got the signal from. It's called Parkinson's law, you know, the proliferation. And none of these things have chloroplasts or mitochondria. The current phrase, the elephant in the room, is inadequate to describe, you know, the lack of attention to what really matters in living cells, which is where we get our energy from. What's important we get right and what's dangerous we get wrong. It's just too much. And chemistry becomes surreal because they can talk about phosphate group transfer but then they have a little arrow here saying makes cardiovascular disease, or senile dementia is the product of this chemical reaction. And the only time they ever think that information comes in from anywhere other than the outside of the cell is when they have things like UV damage. And inside that cell, oxygen is being consumed at a breathtaking rate. ATP is being made, for one of us, as Peter Rich calculated, at 85 kilograms every day. Okay, this is like living next to a nuclear reactor and concentrating on arranging the furniture in the cafeteria. Okay, no mitochondria, no chloroplasts in this. Green box primer 3. Kitchen physics, car, we've done this, is way to really work. Just memorize that if you didn't already know it, it'll be a test later on. Light is radiant energy, electricity is a flow of electrons. Light makes electrons flow, we saw that. That's electricity. Light makes electrons flow. That's redox chemistry, that's where we get all our energy from. Electricity, like light, is physics and chemistry and biology. And we come now to a demonstration of a very important phenomenon. And at this point, I call upon my assistant Sophie. Because I have a slightly more interesting. We have light 1 and we have light 2. And those of you who didn't get the sneak preview earlier on, what I have here, just hold on 1 moment. I have a pair of photovoltaic cells, just like this. I've set them up behind coloured filters, just like this. And the lighting, I need somebody else to do the lighting. I can't do everything. Just like this. You can see coloured filters. That room goes white here. And what the coloured filters do is select a proportion. No, please. Smile. Well, it's very important in oral lecture. A deep thinking philosophical person. Sorry, that was very, very complimentary of you. And what we have is another little electric motor, but it's connected to 2 of these cells, connected in parallel. When you connect cells in parallel, you don't add the voltage, you add the current. Okay? Because they're in parallel, the rate of flow of electrons through one, from them both, is equal to the sum of the rate of flow of electrons from 2 of them. Will you switch on light 1? Sophie, please. Right. So, one of these cells, which is absorbing red light because it's behind a red filter, it's this one, is sufficient to make enough electricity to drive the motor, to drive the fan. I don't think, again, it was one of those signal transduction components that put that lecture in these electrical. Remember the lecture here, you can see everything that you say, no, you will have a lecture in there. Anyway, will you switch off light 1? Will you switch on light 2? Okay. So they're connected in parallel, so it doesn't matter which light you use, one of those cells will get it. Same. I gave you the trash rehearsal. You must listen. Switch off light 2, please. Now what I want to do now is reconnect those 2 cells. Just stay there. Very low. As I said, a parallel connection. And I'm going to change it to a series connection. Just like the series connection when you put batteries in a torch, you connect positive to negative, and negative to positive, so you can add the voltages. Your 2 1.5 volt batteries will give you 3 volts. And this really is asking a lot of spur of the moment stuff. Any qualified electrician is very welcome to come and verify for yourself that that is no series connection. Would you now switch on light 1? Would you now switch off light 1? Yes. on light 2? Would you now switch on light 1? Okay. It's a series connection. You add the voltages. You make more voltage than you would get from one of them, or from both of them connected in parallel. And you need the two lights to get enough energy to turn them over. It's called enhancement. The effect of one light enhances the effect of another. This was a phenomenon in photosynthesis described by Emerson and Arnold and studied and wondered about for 15 years before a biochemist called Robin Hill, came along and said, the Z scheme explains this. Thank you. Is this the lights on? Sophie will be returning later in the lecture. My popular request. So, this is it. We've said it before. They're connected in series. Brilliant. Wonderful. Explains this enhancement effect. It explains the redox potential of the electron carriers. But why are there two light reactions in photosynthesis? Because they must be connected in series. Why could they not be connected in parallel? Why do you need two anyway? What if they were connected, if they were connected in parallel? This is photosynthetic electron transport is not described by Ursula. You can't add the voltages, you can only add the currents, so you get a great current flowing through, but you don't generate a strong enough positive charge to oxidize water. So you haven't got anywhere the electrons to come from. Not in this solar system, in other words you might. And you really can't get low enough, negative enough, to reduce this final electron acceptor. You can do it a little bit. And in fact there is evidence in reports in the literature that you can get oxygen production and a little bit of NADP production from one photosystem, and indeed you can, but it's so slow as not to be worth writing home about. And energetically it doesn't work at this terrific high throughput rate that we require. If you had the two photosystems working in parallel up here, sure you could reduce NADP, but where would you get the electrons from? Certainly not in water, because you would generate enough of a positive charge to be able to get the electrons to the in the first place. So the parallel Z scheme is a non-starter, and the series Z scheme is a starter, and that's why. Why are there two light reactions in photosynthesis? Because they must be connected in series, therefore their voltage is the sum, therefore their currents are equal. Okay? You can't throw away electrons or produce anything behind all that. Therefore, the photosystem's rates of energy conversion must be made equal. Therefore, their activities and quantities must be regulated. How are these quantities regulated? By means of a signal in the electron transport chain going back to the genes to say we need more of this one now and less of this one, or vice versa. And what is that signal? It's in a paper we're publishing shortly, and it suits the broadcast sensor highlands. And without this, you would not be able to regulate a photosynthesis that requires two light reactions connected in series. So, we go back to Dr. Fogg, Professor Fogg, and his cyanobacteria. I'm going to turn very briefly, because I'm aware that time is moving on, to an entirely different sort of green box. This is a picture of a poor, frightened pair of polar bears, frightened that they might have to swim in that nasty cold water. I think this is a hot air blower put there by Greenpeace activists who wanted to take a photograph. I don't see quite else how you could get in sort of perch. I suppose they've been brought in by helicopter or tranquilized and ready to wake up or something. This is going to get slightly, I mean, I will make enemies here, I'm aware. What the hell is going on in electric waste? Here's another one. It's petrified, really. I mean, the thing's never been in water before, let alone cold water. And the last Arctic ice sheet is about to disappear, and all it can do is leap for safety off this one onto this one. But this one's going to melt soon, so really it's going to have to swim and drown, because that's what's probably going to do that. Now, Greenpeace. There are lots of things about Greenpeace I will support, sign up to, and enthusiastically support. I think killing whales is completely stupid, as well as cruel. They shouldn't be done. We ought to let the stock build up. So that's fine. I salute them for that. I don't salute them for this. Number one, this graphic says Greenpeace protest in Denmark. It isn't Denmark, it's Helsingborg in Sweden. I can tell you this, I've been there. That is Swedish. It says no to GMO swine. You're not just a swine, you're a GMO swine. Or pigs, as you would say. And what they mean, actually, we have already, I've got news for Greenpeace, we've already genetically modified pigs. We did it by artificial selection, by breeding them for thousands and thousands of years. And let me tell you, a domesticated pig is altogether a less of a dangerous human than a wild cousin. When you leave them alone, they're much more dangerous than if you take control of them. That's number one. And the other thing is, it's not about genetically modified pigs at all. It's about feeding pigs on soybeans that have had a gene introduced to make them resistant to insect attacks. So you get more soybeans and more feed for the pigs. And so you grow the pigs in some totally mystical way which defies my understanding completely. Andy Cumming surely agrees with this. The genetically modified soybean transfers its genetic modification even after all the proteins and nucleic acids have been digested in the pig's stomach into the pig. So I don't know what on earth these people are talking about. This, I think, is tasteless exploitation, and I have no time for this at all. This poor little child from Finland has been picked up and put down in the remote Cape Desert to support the legend global warming will affect us all. He's the wrong colour, and all the makeup that gives the impression of terminal distress, I think, should be taken away. Perhaps I'm missing something, but I think that is cynical, heartless, callous, and taking us all for fools. And what you make of the iconography of this one, where the animals created is a mutable species going by two to visit President Noah to ask him to sign the Kyoto Treaty. I really don't know which branch of fundamentalism and updatism refers to, but it must be someone who thought America is the end of the Soviet War. Terrible peril if we just believe or something like that. So we're going to go for that. And the news is we've been here before. This is why it's a miracle I still have this in my office. January 90, this is one of the benefits of being old. It cost 25p, which we only get used to saying at that time. It was 5 shillings. It was 5 shillings. It's Blueprint for Survival, Volume 2, Number 1, Man and Environment, Quality of Life. A statement of support. Remember Simon, Sir Frank Fraser. I used to sit around and say, what a wonderful man. And all my people I read, Sir Julian Huxley, who incidentally invented the selfish gene before Richard Dawkins was out of short trousers, not even in short trousers, he just didn't give it a 5-6 name. And so on and so on. I mean, all these great, admirable people, including John Arthur, who's a professor of zoology from California, and told us about ticks a lot, and so on. So they're giving their statement of support. This is where they're going. Do not make no mistake. We thought at the time this was not political, it was political. But many people explained to me, I wasn't from the far right, not at all, never have been. So when people said, well, this would be joy to the far right, I thought, shut up, you're not what you're talking about. But such a movement cannot hope to succeed unless it has previously formulated a new philosophy of life, where goals can be achieved without destroying the environment, and a precise and comprehensive program for bringing about the sort of society in which it can be implemented. Let's put it and Teddy Goldsmith, it helps to be, if you're going to launch a political movement, to be extremely rich, or to have an extremely rich brother. And one of their sons is still doing it, I understand. Is that no relation to Robert Manning. And this is what they predicted, 1972. I bought it back from the newsstand in London somewhere, charity twice, I think, for 5 shillings, if you call it still. And world reserves of crude petroleum at an exponential rate of consumption. So these are the reserves, starting from 1950. We're here, 1972. You can see they're going down, and they're going down, and nothing at all in the year 2000. And you can see they've actually put that intercept as their objective. And this is the production in billions of barrels a year, or demand that's going to be produced. And the whole thing's going to crash in about 1975, as Paul Ehrlich said, when the cost of crude oil is going to rise to a height where it will be unsustainable. The two curves will cross, and by 2000 you'll have eco-catastrophe, social disruption, and the end of the world, because nobody will have any energy to do anything. And it was wrong, right? And here's some more of these sort of Damocles type angles here. One of their other predictions, mineral resources, static and exponential reserves. I look very, very carefully. Whenever I see a graph, I want to know what the axes are. Is anybody else pedantic in that way? And they've got units to them. So they've got a bar graph with numbers in brackets, and all the number of vital elements for human ecology and industry, and so on. And they're all pretty well going to disappear by the year 2000, except for really abundant ones. You'll see now, in 2008, we've already run it with gold, silver, copper. Oh no, we've got probably just a little bit of copper left. Mercury, dead lost, that all went in the 1980s something. Lead, platinum, tin, tungsten, you've got a few years of tungsten. It's all nonsense, you see. And it says, please see for explanation, please see. I can't see from here. Key, page 41. I went to page 41. Here it is. They just write it all down again. Doesn't explain anything. As can be seen from the chart on page 7, that's the chart on page 7, at present rates of consumption, all known reserves of these metals will be exhausted within 100 years. That would be in 2072, with the exception of 6. Aluminium. Christ, you can't exhaust aluminium to turn into earth crust. Try to exhaust silicon or something. The idea is insane. However, if these rates of consumption continue to increase exponentially at the rate then until 1960, then all known reserves will be exhausted within 50 years, so they're trimming it back a bit. That would be until 2022. So, folks, hold on to your copper. The price is going to go up, with the exception of the total. But it didn't happen. It didn't happen. That's not to say that the Greenpeace lot are wrong now. The ecologist people could have been right and just got the time frame a bit wrong. But I think there's something about people that they just like to worry, you know. And I think there's a wrong model behind all this, which I think they're trying to do. What are we going to do? Where are we going to get the energy from? Global warming is correct, I think. The Arctic ice sheet is getting smaller. The polar bears are 100 meters north, at least. I can't understand why we don't introduce polar bears to the Antarctic, actually, so they can eat penguins. Satisfy the old joke. Maybe happy then, so sure. But here, if we're going to get energy, the non-polluting, non-CO2 producing energy sources that are toted. Here is 660 kilowatt. You know what a kilowatt is, it's 1,000 watts. And you know what a watt is, it's a joule per second, so it's quite easy. Wind turbine on the Dwarf Peninsula in Wisconsin. Looks very nice against the setting sun or rising sun or whatever it is there. 660 kilowatts is not even beginning to think about the problem. 660 kilowatts is about 100 electric cookers. Okay? So you have this thing hundreds of feet in the air, making land for many, a large distance uninhabitable, giving you wind damage to the topsoil, so you can't grow anything after a while. And all you get is this is usually 660 kilowatts. Meanwhile, in North Wales, the Ulva nuclear power station, Greenpeace, of course, are against nuclear power stations, you know this. Sits there and it's generating close to 1 gigawatt of power from two Magnox reactors. That's a lot of power. That's worth thinking about. Neither of these produce CO2. There's another solution. Carol and I had the good fortune to visit New Zealand. This is a river we passed. Devotees of Peter Jackson's version of Lord of the Rings will recognise immediately the Great River Anduin. It's here. We went past here and just happened to come across the Clyde. Here it is again, high magnification. The largest hydroelectric dam in New Zealand is the Clyde Dam, built on the Coosa River near the time of flow. This is in South Island in Otago province. Made of 1 million cubic meters of concrete for the dam, and so on and so on. 70 meters wide, it's not a lot actually, 10 meters wide at the crest, 490 meters long. Lots of statistics there. What does it look like? Looks like that. They built it to have 8 120 megawatt hydroelectric turbines, and they got as far as building 4 and ran out of money, or else the geologist said, watch out, this is an earthquake zone. But they changed their plan. You can see there's pretty space for it here. It's fabulous, really. This is real power. This is 480 megawatts. That's half of the nuclear power station. And they only run it at 400 megawatts apparently for safety reasons to keep well within tolerances. What does it look like? What does it sound like? Does it sound wrong, Paul? No, it's not. Okay. Never mind. it should get to that. There we go. And more. And then suddenly you're looking south and east into central, into the same area. And that's what the hydroelectric plant, hydroelectric plant in South Island looks like. And that's half a nuclear power station. If you'll forgive me, let's go a little bit closer to home and recognise this place because I walk past it to the daylight. It's a clean building. And right now we are, I can't see a thing here. We are here, we're in this building here. That's Margaret Road, that's the Green Bridge. We came from Margaret Tube Station, it's right there. We're going to a reception in a few minutes in Thomas, in the Octagon, which is indeed an octagon, as you will see here. And what can we do about power then on that scale in the United Kingdom? And I really am going to take you home now, I hope you don't mind this, this is a bit of indulgence on my part. Where are we going? Away from the Thames estuary, heading west, ladies and gentlemen, to the Severn estuary. The Google movie would have been better, but I did it in still frames. We've only got so much time. And what we could do is think about putting a tidal barrage between about here and about here. Two islands, the Mendic Hills go across the Severn estuary. They reappear above the surface as two islands, discovered by the Vikings, or at least named by the Vikings as Flatholm and Steepholm, which, in my opinion, is quite sufficient reason for burying them in concrete. But that's a personal view. And here they are. And if we took the Mendic Hills, close to where Vanessa, I see at the back, where Vanessa comes from, and Penarth, not far from Newport, I come from somewhere around here. The beautiful River Severn goes like this all the way up into North Wales. The Thames is, it's nice, I like it. It used to be a tributary of the Rhine, you know, before we separated from the continent. Whereas the Severn has always been the Severn. And on the left, we have the Welsh. The Romans, it's a disputed border territory. The Romans pretty well gave up by the time they got here. They built a nice fortress with amphitheatres and so on. The Anglo-Saxons gave up by the time they got here. It's rumoured that there was a man called Ursula who built a dike to keep the Welsh out, but whether the Welsh built the dike to keep off, and on the left we have the Welsh with their demonstrable genetic superiority at rugby union football. And the idea is you simply build a bridge across the estuary from there to there, and that's Ladderknock on the Welsh side, which I know and it's misty and mysterious and wonderful. And so we have a choice. What do we do? 660 kilowatts. It's pathetic. It's window dressing. It's really just public relations. They're not even on the map. We have a nice, tidy, sensibly managed nuclear power station, which is run by people who think, rather than people who obey orders, which is the Chernobyl. Or we have the Severn barrier, which would produce about 8.3 gigawatts, but untold ecological damage, of course, because the tide goes up and down by 15 metres. It's the second highest tidal reach in the world. Awful lot of water, awful lot of power here, but it's clean, it's free. Well, it costs 15 billion to make it, but once you build it, it runs itself. And there you have it. Or maybe we could harness the power of photosynthesis. Where's Professor Melody, who has just organised a magnificent bid for Queen Mary and other co-workers for an immensely small sum of money in the great scheme of things, to see how high cyanobacteria could be used to use light energy to make hydrogen as a fuel. Well, that's a very good idea, actually, a very good idea indeed. But let's do the math, as the Americans say. What would you get? Now, I estimate 10 watt per square metre in Britain. In the equator, you get more. That's 100 kilowatts per hectare. So if you want, so this thing, pathetic as it is, we still need 100, 66.6 hectares to get equivalent amount of power from photosynthesis. If you want a gigawatt, if you want a nuclear power station's worth, from cyanobacteria making hydrogen, you've got to find 100 square kilometres from somewhere. In Britain, I don't think you get much votes, really. I hope the principal is not going to try to tell Gordon Brown that you've got this great idea all we need is 100 square metres of photosynthesis over central London, because it would look like that. That's where we are now. That's 10 kilometres by 10 kilometres to make one nuclear power station's worth of energy by photosynthesis. Not going to work. People aren't going to go for that. It might work. However, in Australia, which is mostly landmass, which gets a lot of sun, which is not doing a lot else. And there's this thing called the Sahara Desert. So in terms of the ecologist living in a converted growing his own organic vegetables and running a Toyota Prius and feeling virtuous, you can't solve the problem this way. But in terms of global, sustainable, non-polluting, free energy from sunlight, it might be worth thinking about. Back to the cyanobacteria, nearly there. Acknowledgements. This is an inaugural lecture, not a valediction, therefore I cut out dozens and dozens of people, including good friends here, who I will acknowledge one day when I retire. Please come back in 30 years. Okay, but here's Conrad Molyneux, who by some amazing stroke of astronomically improbable fortune, is now a colleague, but he was once my PhD student in Leeds. He's doing rather well. Let me say, he's Professor of Microbiology at University of London. As I say, he's putting together this bioenergy board amongst other many issues he's doing. Michael Harrison, my second PhD student. I've never heard of many PhD students, but they all do wonderful things. Mike's doing a Wednesday seminar at noon and is very lecturely on a date of March, which slips my mind, my memory for a moment, so he comes back to that. He went on to work in the University of California, Berkeley, with Tasso Melis, who is the beloved advertiser of green energy for OD motor cars. If that's not a piece of cunning brand positioning, I don't know what it is. But that's Mike, and he's doing great stuff. And Nicholas Chiorowicz, who's my PhD student in Leeds, he went to do a postdoc in America and joined a .com genomics company called DoubleTwist.com, which the bubble burst, and he wound up working at the Scripps Institute. And then moved to Scripps, Florida, and is now Professor of Computational Biology at the Department of Medicine in the Miller School of Medicine in the University of Miami. So that's pretty good, don't you think so? Christine Sanders from those days was a super technician in the research assistant. Unfortunately, she got married to Conrad and went off and had family and lived in Germany and good things. She's here tonight. Carol's been my constant co-worker, companion, life support system and interface with the real world so far as that's possible. These are the two of them in conversation at Conrad's inaugural lecture last year. Thomas Fanschmidt, postdocs I've had lots. Thomas Fanschmidt has now gone quite a lot bolder. This must have been some time ago. He was my postdoc in London, Sweden. He discovered the redox control of podcast gene transcription in our lab in Sweden. Show you it happened. He didn't have a mechanism. It took Susie to come on before he had a mechanism. Nigel, I've seen you. Where are you? I can't find your photograph, but I have a camera with me. Nigel Holmes, Genetic Section Animal Health Trust. So we were a small group. We ran on nothing. Leeds University were extremely enthusiastic, and we did wonderful things in the all life. Oh, except for Mike. I don't know who he's doing. Sujit, you've heard about before. He's here. He's a Kuhn. He's discovered the Higgs boson of molecular cell biology. We have an ad hoc group that has no formal status. It's just that we have these valued, wonderful colleagues, distinguished people. John Neal had a wonderful website, and I said, why don't we have it here? And he said, okay. I said, okay, you can have my computer. So we made a website, and that's all the significance there is in this. We have links back to the School of Biological and Chemical Sciences, which is full of clever, nice people. I kid you not. It's such a pleasure to be here. In a whole range of things, from chemistry to ecology, and it's marvellous. And Brenda Fake, who left me a fog diagram, and this is the family who've had to put up with all this nonsense. And the glamorous Sophie is going to be my willing assistant yet another time. Sarah drew some wonderful cartoons, which I had to throw last minute. Nicholas helped me set up the demonstration that is yet to come. And this is 15 minutes walk east of where they are, believe it or not. Here's the green box, number 1, the chloroplast, which came from a bacterium. You know now why it didn't gather all its genes. When the worst happens, we didn't need emergency escapes after all. Fire extinguishers, somebody had better check on fire extinguishers. I see no fire extinguishers, perhaps you all escape. When the worst happens, you email central administration, they don't reply, you get an out-of-office automated reply. Somebody entirely different emails you back in 2 weeks after the event and says, okay, we've got some action, and the action you get is that somebody who knows someone in the local fire station says, okay, we've got some work for you guys, and they come out after your house is burned down and put out fires everywhere in your town. Okay? That's what the nucleus does. It doesn't know anything. It doesn't have the information. On the ground, you know where the threats are and you know where the opportunities are, and you solve the problem there and then by being able to use your own fire extinguisher, and that's what the bypass central firing does. You thought it was about to end, but there are no revision notes, so pay attention. Kitchen physics, electricity, electrical potential is a charge difference measured in volts. Electrical current is a rate of flow of charge measured in amps. And power, this is one of the most useful things you can ever know, is equal to potential times current. Watts equals volts times amps. You never put the wrong fuse when you type on your kettle, if you remember that. And what I want to do now, yes, here we go. The next bit is demonstration. We saw the climb down in New Zealand. We're now going to see the full awesome power of 13 watts of clean, free, ecologically responsible energy. Sophie, where's Sophie? Please welcome Sophie. She's feeling rather shy. We have the connections. We've flown the kites. Did I predict the light on? We don't have a kite. And what I want to do is give you some glimpse of the future and how our high energy demand society can indeed be sustained entirely on sunlight. Will you switch on light 1, please? Let's switch on light 2. It doesn't what's happening. Disaster. I was determined that this would learn the first time. The battery's red sick. I know what it is. All along I promised to produce the solar suitcase and prop it up like that. And we still have to do it. We tried this earlier, but yep. You've got to connect it to please light one off. Light 2 off. Both lights on. We'll do these and put your hand in front of one of the lights, just so we know there's no battery, there's no I do the other one. It's quite hot, I appreciate that. we need a coloured filter here. There you are, look at that. And just please stay there. There's another small postscript here. There's a competition. Yours for free. It's a gift if you win the competition. It's 300 milliwatts of clean power, non-polluting, no maintenance costs. It runs forever. Come up front, please. And all you have to do is the educational solar energy kit, which I use to make a little windmills running on photovoltaic cells. And it will go to the first person who raises their hand and answers correctly the following question: what is the projected power output of the 7 barrier if it was built to the nearest gigawatt? Raise your hand. 8 gentlemen there. Please. Well done. That's the green box, that's the energy, that's GE cost. Thank you for your attention. Next one, please, is where we work, the Fog building. We're on the 6th floor, and that's the end of the lecture, and I thank you all for your attention.
Adrian doesn't know what he missed. He should have brought Gordon Brown here. Thank you very much, John. That was fantastic. By tradition, we don't have questions. That is after a lecture, so we don't have formal questions. We do have a reception, and we can ask lots and lots of questions. So, thank you very much.
Thank you.
Incredibly stimulating and a wonderful inaugural lecture. You're all invited to go to the Octagon for a reception. A lot of people know where it is. If you don't know where it is, contact us.